So now let's continue with next type of interaction called the predation. Just as the term implies predation, it mainly involves the what a person or an organism which is predating or feeding on the other one. The when we consider the term predation, the picture which comes across our mind is of course a lion predating on a deer. That is a good example of predation. Now, coming to the microbial world, they also have similar type of predatory movements in which the microbes will feed on other microbes. And the most important example of that is Delovibrio. And we also have various other organisms such as the vampirococcus as well as the Daptobacter. These are all examples of predatory bacteria which have been found in the nature. Delovibrio is a periplasmic predator which will penetrate the cell wall and grow outside the plasma membrane. So if you go to look here, you can see this. If this is a cell, it will penetrate into the what? The cell wall, and it is growing outside the plasma membrane. In the case of vampirococcus, it occurs as a unique epibiotic mode of attacking the bacteria. That is, it's a little bit outside itself, but it will invade the organism. In the case of Daptobacter, it is showing a cytoplasmic location as it attacks the susceptible bacteria. So we'll just go into the details of these three organisms. Now, in the case of Delovibrio, it is an active hunter which is highly motile, and it will swim looking the susceptible gram-negative bacterial prey. You should remember that Delovibriae, it usually attacks mostly only the gram-negative bacteria. Okay, and this one you can see that mammalian cells, okay, mammalian cells are not being affected by these Delovibrio, but the E. coli cells or the gram-negative cells which are growing in the human gut, etc., as well as in the other environments, can be attacked by this Delovibrio. So it will swim very fast, and until it collides with its prey cell, it will be migrating in a very fast rate. And once it sees its prey, what will it do? It will penetrate, it will make a hole on the outer membrane of its prey, and it will enter the periplasmic space. And once it enters into the periplasmic space, it will start producing long filament-like structures that will eventually septate, or it will break to produce the progeny bacteria, and the lysis of the prey will release the new Delovibrio. So when it is feeding on the gram-negative bacteria, okay, it will utilize, of course, the cytoplasmic ingredients of the prey, and it will survive over the organism. Now, you can see that, as I mentioned earlier, the Delovibrio will not affect the mammalian cells, but will affect only the gram-negative cells. And gram-negative bacteria has never observed to acquire any resistance to Delovibrio attack. So, there was an interest recently that why can't we use Delovibrio as a probiotic to treat infected wounds and all that? But trials are yet on the way, and later on might be this could be serve as a what? It could serve as a good agent for treatment of wounds which are being caused by gram-negative bacteria. Mostly the wounds are being get infested by gram-negative bacteria. So there is a hope that probably this Delovibrio could be used further on for the treatment of the wounds also. Another one, as we mentioned here, is Vampirococcus and Daptobacter. They also kill their prey and they gain entry in a less dramatic fashion. What Vampirococcus does, it will attach as an epibiont. Epibiont, it is a biont means which is being having some biological relationship with the organism, but it is considered to be epi because it is on the surface. On the surface of the outer membrane it gets attached, and after that it will secrete various degradative enzymes that will result in the release of the prey's cytoplasmic content. Okay, so in the case of Daptobacter, that will penetrate into the prey cell and then it will consume the cytoplasmic contents directly. I guess you got an idea of the predatory bacteria and what is the use of all such predatory bacteria. There are different benefits which we consider. Now, for example, the simple ingestion and assimilation of the prey bacterium can lead to increased rate of nutrient cycling. Okay, so the cycling of even what happens, the organisms, the microorganisms need to be cycled back into the microbial loop. So for what is that? For to cycle these microbes also, like of course bacteria feed on other matter. This same bacteria has to be degraded and again it has to come into the pool. For that, these predatory bacteria can serve and various protists. Okay, in the case of protists also, the ingestion as well as the short-term retention of bacteria is needed in the case of various protists. And so what happens, they when you need only a limited supply or limited retention of these bacteria in the rumen, what happens, these can be predated. Okay, and you can see that methanogenic bacteria will contribute to the health of the ciliates by decreasing the toxic hydrogen levels by using hydrogen to produce methane, which then is being passed from the rumen. So to control the bacterial the bacterial colonies, the amount of bacterial colonies in rumens also, the technique of predatory organisms can be utilized. Now, methanogenic bacteria, they are beneficial for the uh, for the ruminant animals because the hydrogen which has been produced in the rumen of these ruminant bacteria, if lots of hydrogen accumulates in the ruminant stomach, that what will happen? Hydrogen gas filled or hydrogen balloon. Do you remember what will happen? You can just imagine what the cow itself might go on top if it is filled with hydrogen as such. So there should be some mechanism by which this hydrogen has to be converted. Otherwise, the cow will be flying on the air. Okay, so in such cases, there are methanogenic bacteria which will convert the hydrogen which has been produced in the ruminants to methane. But beyond this level, when methanogenic bacteria have to be, have to be controlled, a predatory role will come there and it will control the methanogenic bacteria. And at this, so in order to maintain a balance between the various organisms which are being involved in it, we can use a predatory bacteria. And at the same time, a protective as well as a high nutritive environment is being given for the particular prey. Now, for example, the protist ingests the gram-positive bacteria Legionella and protect this important pathogen from chlorine. Okay, which is often used to an attempt to control Legionella in cooling towers as well as the air conditioning units. Okay, so this in some cases what happens? In order to protect certain gram-positive bacteria also from high nutrient environments and all. Okay, in order to maintain a nutrient environment, in a protective environment, some bacteria might be predated inside. Now, for example, in cooling towers and air conditioning units and all, chlorine is being found to be inhibitory to the organism. So what will the protists which have been found over in that environment might sometimes predate onto gram-positive Legionella and protect it from chlorine. So not only the case of nutrients, okay, but some cases also from the product for as a protective measure also the predatory mechanism can be used. So these are the two benefits of predation as I've explained here. I hope this is clear to you and we come to the end of this session. We will be continuing the next session. Thank you for now.